Okay, can everyone hear me? Yeah, I think so. Okay, so first of all, I'd obviously like to thank CCP4 for organising this event and also giving me the chance to present some of the work that we have been doing in Liverpool recently. And today I'm going to present to you ConKit, a Python interface to contact predictions. But before I talk about ConKit itself, I think not all of you will be familiar with the idea of contact predictions, and in particular how this can be useful to you as structural biologists. So in the field of contact predictions, a contact in general is simply defined as having two residues in spatial proximity. And this does not necessarily mean that these residues have any kind of functional relationship, but purely that they, as it says, are close to one another. And when we do contact predictions, we're trying to observe a covariance signal. And when you take the following two amino acids in the middle of this image, and only a single amino acid or a single residue of that pair would change, as you can see on the left, then if those two residues are relevant in the structure or function of this protein, would most likely going to be selected against. However, if you go from the center of this image to the right, where both residues change simultaneously, then so change simultaneously here, then if this change gets accepted, this will ultimately lead to what we're trying to observe, this covariance signal. So the change from here to here. And in bioinformatics, when we do contact predictions, we can choose almost one of two routes to do exactly that. We can either choose evolutionary coupling analysis methods, or we can use supervised machine learning algorithms. And the key difference really is that evolutionary coupling analysis methods use sequence alignments of many homologous sequences to try and detect or observe this covariance signal. These methods in general produce very highly accurate contact predictions. However, as you can imagine, they do rely on having sequence homologs available. And I will talk about this a little bit later on. If you use supervised machine learning algorithms, then you do have some kind of lower accuracy in your contact predictions. However, you do not require any sequence homologs. So in cases when you work with, for example, a protein target, which doesn't have many sequence homologs available, this might be the method that you want to use. And some of the latest data that we sort of have in Liverpool show that these supervised machine learning algorithms actually produce accuracy nearly as high or even sometimes higher as evolutionary coupling analysis methods in cases of transmembrane proteins. So these methods might be particularly useful for transmembrane proteins in your studies. I guess the slide that most of you will be interested in is how can you use bioinformatics and contact predictions in your structural studies. And this can really has a wide range of different applications. It can be something as early as characterizing your target when you, for example, want to define your domain boundaries more precisely. At the same time, when you're at the stage of molecular replacement, you can use contact predictions to, for example, detect any conformational change that your protein search model, for example, requires to solve that structure. In Liverpool, we've been working with contact predictions in the context of search model building, where we've used them as distance restraints during ab initio modeling. And there have also been some studies that showed you can use contact predictions to also do map interpretation in cases where you have low resolution density maps and you can't assign the sequence register of your search model. If you do have already a structure and you want to do some more structure interpretation, then contact predictions can also be very, very useful if you want to, for example, identify functional sites. And I'll talk about an example in a second. So as I mentioned, in Liverpool we're interested in creating search models for molecular replacement, and we do so not by using sort of your close homologs, but more using ab initio modeling or distant homologs as starting points. And what you can see in this figure is an ensemble mapped against or superimposed with its crystal structure in the cartoon at the background, where the ensemble is composed of these models made up of the tubes. And this one here on the very left was create, or this ensemble is made up of models that were used with simple ab initio modeling algorithms that didn't have any contact predictions included. And the number of models you have within this search model can usually be correlated with the quality of your modeling in the first place. What you can see here on the right, in these two figures, is exactly the same target. However, at this time when we did ab initio modeling, we actually used contact predictions as distance restraints to sort of guide the modeling algorithm and limit the conformational space it had to search. And as you can see, these ensembles are much richer in terms of the number of models they contain. And when we started running them through molecular replacement, these two actually led to structure solutions, whereas this one didn't. So contact predictions can really be beneficial in this context. I also said that you can use contact predictions to identify functional sites, and this is a slightly different approach to using your contact information. Most of the time, your contact information is used as looking at the predicted contact pairs, whereas here, we actually analyze each residue and its network of predictions according to their covariance score. And those that have the highest overall covariance signal tend to be found, those residues, in functional sites or protein-protein interfaces. So this information can, again, be very useful for you to identify, for example, functional sites in your studies. So what was the overall motivation to create or develop an interface to all of these contact predictions? Well, in general, contact predictions are getting better and better, and this is partially because as we progress in time, there are more and more protein sequences deposited in databases such as UniProt. At the same time, when you look at this contact map here, where you have residues on X and Y axes, and each basically dot corresponds to a predicted contact pair, then what you can see is that this is an old statistical model that has been used for many years, but has been updated sort of about 7 years ago, or changed, and with that, the accuracy of your contact predictions has become much, much better, and we can now actually use this information because we can filter out a lot of the noise, as you see here at the very top. At the same time, there are quite a lot of different contact prediction algorithms, as well as algorithms to obviously construct your sequence alignment in the first place. And when we started working with contact predictions, what we'd found was that it's incredibly difficult to actually use all of this information from different sources, because you would almost always have to write some kind of code to handle the data and analyze it. At the same time, contact predictions and the whole process is very much aimed at bioinformaticians. So unless you have some kind of familiarity, for example, with the command line and how to install and use software at that end, you would really have difficulties exploiting the most of contact predictions. So the result of this is what we call the Contact Prediction Toolkit, or ConKit. And ConKit itself is an application programming interface, or in short, API. And for those of you who are not familiar with what an API really does, I've decided to choose this sort of relatively simple analogy, I think. Take the following example. You go into a restaurant and you're presented with a menu of options. Now, you have no direct way of communicating, for example, to the kitchen what food you would like to have. At the same time, if the kitchen knows what you would like to have, they can cook your food. But the cook normally doesn't bring out your food directly to the table. So what do? In a restaurant, you talk to a waiter who basically can fulfill the wishes or the orders that you have on your place. And the waiter in this case can be put equivalent or on the same level as an API, because it provides for you one simple interface that you communicate with in a restaurant, and he knows how to communicate with every other party within the restaurant to fulfill your requests. And ConKit does exactly that in contact predictions. It basically is made up of 3 sub-packages in terms of its code, and at its center really lies the core package. And this core package will store all your contact information as you read it in, and will allow you to access the information, it will allow you to manipulate it, and it will also allow you to analyze your information as you wish. Normally, you wouldn't construct the data model that it creates, though, from scratch. You would much rather use one parser that comes within the input-output subpackage that will basically allow you to just provide the file and tell ConKit what format it is in, and it will construct all of this for you. If you want to run or create contact prediction pipelines yourself, we also provide a set of different command line application wrappers, so you could write entire pipelines purely in Python without having to know or having to worry about anything else. In general, you can interact with ConKit in two different ways. If you're familiar with the Python programming language, then you can access the full functionality of ConKit via, for example, a Python interpreter or a Python script. And if you're not so familiar, or you want to just quickly run certain analysis, then we have also written a set of scripts that you can invoke from your command line that will basically expose the key functionality of ConKit with very simple sort of instructions. And instead of me going into more detail about basically how each of these different subpackages are made up of, I thought it would be far more useful to actually show some examples of how you can use ConKit in your own studies. And all of my, or pretty much all of my examples are structured in the way that you always have two boxes, black boxes, and at the top it will show you how you can basically run a script in ConKit, and at the bottom is the equivalent Python code to do exactly the same analysis. And this particular example here is the analysis of a multiple sequence alignment. And I said earlier, when you do evolutionary coupling analysis, the alignment of with many sequence homologs is very important. And this script will basically compute for you the best descriptor for that alignment that will be indicative of how good your contact predictions will be. And it will compute something called the number of effective sequences. And the number of effective sequences basically describes the number of divergent sequences within your alignment at some sequence identity threshold, in this case 70%. And those sequences will actively or significantly contribute towards your contact predictions. So when we take this example here, we have the PDB target 1 EAZ, where I constructed an alignment, and analyzing it at 70% sequence identity, we basically have in general over 51,000 sequences, but only 6,109 of those will significantly contribute to your contact predictions. Now, some of you might wonder, what is the kind of number that I'm looking out for in this particular case? If you have more than, I would say, 100 to 200 effective sequences, your contact prediction using evolutionary coupling analysis will almost certainly give you very good results. If you have less, I would recommend you use supervised machine learning algorithms, and the reason for this is just that your contact predictions may contain some useful information, but it will almost certainly get lost within the noise of the rest. A second example where ConKit is very useful, and I think the only tool really allowing you to do all of this out there, is converting different file formats. So I mentioned earlier, working with contact prediction means different file formats for almost every server out there. And this script basically allows you to convert any of or over 15 different contact prediction file formats interchangeably amongst it. And it also allows you to do the same for some of the sequence alignment formats. Now, there's obviously a lot less sequence alignment file formats here, but the reason for this is purely there aren't many more that are typically used within contact predictions. So this seems to be enough for now. But this list will obviously expand and extend as there are more prediction file formats out there. Now, if you do contact predictions, for example, on a server or locally, what you normally end up with is a file with your individual contact pairs. And this will look something like this, where you have residue 1, residue 2, and some kind of values that basically describe this contact. But this in itself is not, I guess, too informative when you just look at a list of numbers. So a very common representation or visualization of contacts in this field is using a contact map. And that looks something like this, where you have, again, residues on the x and the y axis, and along the diagonal you have a mirror image basically of the lower and upper triangle. And what you can do in ConKit is you can basically provide your contact prediction file, and you can visualize your contacts in this fashion. Now, if you have some structural information, such as you have already a crystal structure, and you would want to identify functional sites, yet you're not sure how good your contacts are, then you can plot exactly the same, but with reference to this crystal structure. And what this script will do for you is it will color all true positive or correctly predicted contacts in green and all false positive contacts in red. And it will also show you all the contacts it extracted from your reference structure, so you know where you would expect the contact to be. Now, the final example, and I said that most of my examples actually have the Python code as well, but this one doesn't, and the reason is this is basically a script that allows you to run contact predictions locally. If you may work with some sensitive data and you don't want to submit it to online servers, but you are happy to, for example, install a sequence alignment algorithm such as HH-Blitz with its database and a contact prediction algorithm CCM-Pred, then you can use this script and just provide the database and your sequence file, and you will be able to basically, or this script will predict contacts for you and give you a final contact map with your predictions out at the end. Now, if any of you would be interested in what the Python code looks behind this, you're more than happy to talk to me, but it's too much, I think, to fit on this slide. So, yeah. If you want to use online servers, that's also not a problem. There are plenty of contact prediction servers out there, as well as sequence alignment generating algorithms. So on the script I've shown on the previous slide, you can also just provide it with an alignment if you have the prediction algorithm installed. Alternatively, if you want to just run everything online, you can just basically paste your sequence in some of these online servers that are out there, and you will get a contact prediction file, and sometimes with some analysis already done for you. And you can, if you don't have any analysis, provide this to ConKit and analyze it in this way. So the last sort of bit I want to talk about is the integration of ConKit into other applications. And I'm also part of the AMPL developers group, and we were particularly interested in exploiting contact predictions in the concept of AMPL. Now, for those of you who are not familiar with AMPL, it's basically a pipeline for unconventional molecular replacement, which means we use, for example, ab initio modeling or single or distant homologs to create ensemble search models that we will trial in molecular replacement. And this is often applicable or useful in cases where you don't have any homologous structures available. And what we've shown last year was that when we use, as I mentioned earlier, contact predictions as distance restraints during ab initio modeling, the quality of the models will enhance significantly, and this will basically allow you to obtain or have a far wider tractable target range using our approach. And with the full integration of ConKit within AMPL, and this will be with the next update of AMPL, you will be able to provide over 15 different contact file formats directly to AMPL without having to worry about converting them in any way. It will also allow you to, or AMPL will, using ConKit, provide you some kind of feedback of the quality of your contact predictions and how it will. And we will also aim to use contact predictions to further enhance our search model building pipeline with some ways such as scoring models based on how they satisfy the contact predictions. If you would want to use AMPL with contact predictions, it's very simple. You can just provide on the command line the -contact file flag and the path to the file. And if you much rather use the CCP4 interface, then we have a restraints file option here. And these restraints files are not necessarily contact files as I've been describing them so far, but rather already converted formats for specifically Rosetta modeling. But if you use the Gremlin server, where the link is also in the interface, it will basically produce those restraints for you that you can just paste in here if you want to use AMPL with contact predictions. Other works that we are exploring is contact visualization plugin for CUDE, CCP4MG, and PyMol, and this basically relies on ConKit to handle any of the manipulation of the contact predictions. And what an early beta version that I've created will basically allow you to do figures or visualize contacts against your structures, as you can see on the right in this on this slide. A second example, not necessarily applicable probably to all of you, but is we're also working with the developers of Tempe in the CCP4 package, and they're very interested in using ConKit to handle distance restraints within their package. And hopefully this list will obviously expand as ConKit is available and people and developers will integrate it into their software. So what do I conclude from here? Well, in general, ConKit is a very versatile yet easily accessible interface. It will basically provide you with a link between the bioinformatics and the contact predictions and all the applications that contact predictions have in structural biology. If you're a software developer, it will basically allow you to access and handle contact predictions within your software very, very easily without having to worry about all the overhead of different file formats, etc. And hopefully, with, as I said, or the integration of ConKit into other applications, a lot of these analyses will become easier to use for you as structural biologists and will become more part of your sort of normal routines of trying to solve your problems. And with that, I would like to thank my supervisors, Dan Rigdon and Olga Mayans. I'd also like to thank the guys working in our group, Jens, Ronan and Adam, for helping to develop and test ConKit. I would also like to thank, obviously, my funding body, CCP4 itself, and the BBSRC, as well as CCP4 for their early support of ConKit and the University of Liverpool, where I'm based. If you are interested in ConKit or if you're interested in AMPL, then we invite you to come today to the lunchtime bite session on AMPL. We'll split the session depending on how interested people are in AMPL or ConKit, and we will show you, for example, how you can use it and how you can exploit it almost from today onwards. And if you would want to look back at the slides, or if you would want to just have a look at the documentation of ConKit itself, then I would direct you to the link at the bottom which reads conkit.readthedocs.org, where all of this information will be available to you. And with that, I'm happy to take any questions if there are any.
Okay, so it might be a bit out there, but have you tried using this with, say, low complexity regions that don't have necessarily any canonically folded domain to look for contacts within those regions at all?
So, in general, in terms of your question is more of a side of applications and how you can exploit contact predictions. I'm not entirely sure whether anyone else has done it. We haven't personally. But certainly if you, for example, would use ConKit and look at your predictions when you have a plotted contact map, it would allow you to see, for example, contact dense regions very easily. And this is, I think, very similar to defining domain boundaries, because contacts are either found intramolecularly or intermolecularly, and the regions in between should only have very few, if any at all.
How computationally complicated is ConKit, since you might run it online or offline?
So ConKit in itself is actually not online. It's only offline, but it's well, personally, I would say it's relatively easy to use, but I've also written it. I guess if you use the script, it's not any different than using any other script. I think it's relatively easy to use, and if you're familiar with using the Python language and packages, then again you should be fine, I think.
The CPU time, how much is required?
So ConKit in itself doesn't use any as such, because it's only an interface allowing you, for example, to convert files. So it's basically reading the file and writing the file. There are some aspects, like, for example, calculating the number of effective sequences, which will take some CPU time, but that's dependent on the size of your alignment. So it can take from, if it's a relatively small alignment, I would say, 10,000 sequences, it can take not even a second. And if you have 60 to 100,000 sequences, it can take a few minutes, depending on the CPU computational power you have available.
Just a simple. It's me.
Where?
Me.
Yeah. Hello.
A simple question. You showed there were 15 different web servers.
Yep.
Have you considered? Does ConKit automate submitting your data to those web servers and getting the results back? And have you considered doing that?
So the simple answer is no. We haven't looked into this at all. In general, I think it's a very good idea. It's probably even possible, but we haven't looked into that kind of aspect at all.
All right. I don't see any other questions. So thank you, Felix. And then the next speaker is Tim Grüne on electron crystallography and the progress of